Okay. All right. So this is in essence a primer on the different techniques that you can use in studying carbohydrate-protein interactions. Obviously, there are a large number of techniques, and each have their own advantages and disadvantages and many other things. We are going to have a discussion on what do these techniques actually mean. So this is a leader sort of session. Classification. That doesn't mean that this is rigid and absolute and hard and fast, but for most part we have quantitative methods, fluorescence and surface plasmon, resonance and ITC and NMR, and all of these techniques are typically fairly quantitative. We actually get hard numbers, and therefore there is a lot of value to it. There are other techniques that are fairly qualitative. People have used qualitative techniques and converted into quantitative numbers. For example, affinity chromatography is a qualitative technique, but affinity chromatography in relation to, for example, affinity with a heparan column has been converted into binding affinities, that this one binds better and this one binds weaker by an X-fold. And we don't really know as to whether how valid that conversion is. Immunochemical methods and things of that sort. In silico methods, modeling and dynamics are quantitative, but I typically like to classify them as an independent class. These are very good methods to understand how recognition occurs, yet these are modeling techniques. So for most part they have to be supported with real experimental evidence, and obviously there are animal models. So what I'm going to do is I'm going to focus on very few techniques, some of the techniques that I think we are going to have training session in the next camp. So this hopefully would also prepare you, and these are the techniques that have been used quite a bit in studying gap binding to proteins. So this is fluorescence, and here is a very simple primer to what is fluorescence. Okay, so we, for most part, have fluorescence because of light release, because of release of photons from the molecules. So some molecules are fluorescent, not all molecules are fluorescent, and that arises because molecules absorb light. So molecules typically absorb light, and the absorption converts molecules from the ground state into something called an excited state. And later on, this energy that has been absorbed by the molecule has to be dissipated in some form. Majority of molecules dissipate it through vibrational means, so increasing temperature, for example. But some molecules dissipate it by release of light. From the excited state, they come back to the ground state, and that is fluorescence. Okay? That is fluorescence. Likewise, we also have a release of light from a different excited state called the triplet excited state, and that is called phosphorescence. And this phosphorescence has not as yet been much utilized because appropriate probes are not many. But this fluorescence is a phenomena that is typically very useful in studying interactions, and there is a big reason for that. So for most part, the absorption is an extremely fast process. It typically occurs within 10 raised to minus 15 seconds, femtoseconds, to 10 raised to minus 17 seconds. It's an extremely fast phenomena wherein the molecule is essentially steady. For most part, the molecule is steady. You shoot light and it basically absorbs, and the molecule has no time to move around for most part. So you don't really see any motion in approximately 10 raised to minus 15 to 10 raised to minus 17 seconds. Fluorescence, on the other hand, is approximately a million-fold slower, can be a million-fold slower. So it takes nanoseconds to really release the energy. And so that nanosecond time frame allows the molecule to shift a little bit, can introduce a small change in the structure of the molecule. It could be a very small change, a few groups moving around. Number of molecules actually change quite a lot. And this time frame wherein the molecule can change can be easily converted into what is happening at the binding level. So that is why you can actually sense changes in fluorescence upon binding. And phosphorescence is actually a much, much slower process. It takes a long time for one of the excited states to move into a triplet state, and then followed by that it takes even longer for the release of light, because this is for most part a forbidden transition. So this can take place of milliseconds to tens of seconds, could be 100 seconds also, that you can see. Once you shine light, that will keep radiating if it is really a good phosphorescent molecule. But we are interested in really fast things, experiments that we can do fairly quickly that allow us to understand as to how molecules are moving around. And what also happens is that in this process there is a change in these energy levels too, and therefore we can actually sense it. So the environment plays a fairly important role. This is once again a primer on what a fluorometer looks like, just a schematic. So in essence, we have a sample in the sample compartment, okay, and this sample compartment is light is allowed to shine on the sample, and light passes through something called a monochromator. The monochromator is the one that you can select appropriate light of appropriate wavelength to pass through. Okay, these are some lenses that help focus the light to approximately the center of the cell. So under normal conditions, imagine that this is not here. Imagine that this is not here. Light would basically pass through, and in the UV spectrofluorometer that you have probably used, you would collect light around here, and you would basically measure the difference in intensity between the two lights, and that is your UV spectrum, right? That is your UV spectrum, and you convert it into absorbance is the log of incident versus what comes out of the at the bottom. In a spectrofluorometer, you don't do that. In a spectrofluorometer, you collect light at a 90 degrees angle. So you can either collect on this side, or you can collect on this side, or you can actually collect on both. There are some spectrofluorometers that allow you to monitor two wavelengths simultaneously, or multiple wavelengths simultaneously. So you can have an L-shaped geometry or a T-shaped geometry. And this is another monochromator. Monochromator means ability to identify the wavelength of the light. So whatever light that is incident here is usually not the light that comes out. There is always some loss of energy. So we measure what light comes out here through a photomultiplier tube, and that in essence gives us the fluorescence spectrum. By varying the wavelength here and by following what comes out, you actually get the fluorescence spectrum, and that fluorescence spectrum is dependent on the molecule, the probe, and the environmental conditions, things of that sort. So it's a very nice tool to study interactions, because as molecules bind, what happens? Usually there is some change, small change, an electrostatic change or conformational change, things of that sort. And therefore, you actually see a change in fluorescence. So it turns out that this nice tool can be very easily used for calculating affinities, hard quantitative numbers. So here is the fluorescence emission spectrum of antithrombin in the presence of several pentasaccharides. So what is done is that we have, this is the heparin pentasaccharide. This is the pentasaccharide that we keep talking about that has the 3-O-sulfate group, which once again we keep talking about. And then these are variants of the heparin pentasaccharide. So this is only 4. This is only 3 from this side. And then this is once again a modified version of the heparin pentasaccharide that has 2 3-O-sulfate groups, 3-O-sulfate here and 3-O-sulfate here. And then we have a tetrasaccharide from this side and a trisaccharide from this side. So we have different combinations, and I didn't make it, by the way. Okay, so this is from a company, Sanofi provided it to us, so we were really lucky to be able to understand how everything works. So this is the normal spectrum of antithrombin, antithrombin all by itself. And the excitation is about 280 nanometers, and you can see that the fluorescence spectrum has emission at 340 nanometers. Okay. So in the presence of the pentasaccharide, or this tetrasaccharide, or this tetrasaccharide, a trisaccharide, or the other version of the pentasaccharide, all of these molecules that in essence contain residue D, E, F, this group gives you very similar fluorescence change. It increases. This is about a 30% increase in fluorescence intensity. Thanks. Okay, so this is about 30% increase in fluorescence intensity. And 30% increase is a good increase in terms of fluorescence. It's a beautiful increase. And you can see that for most part, reasonably similar molecules are able to induce similar increase. Whereas, if you look at this trisaccharide, which doesn't have DE, or this tetrasaccharide, which doesn't have D, one residue change actually significantly changes the fluorescence spectrum. Whereas this tetrasaccharide gives you more, this trisaccharide gives you less. And therefore, those two are actually behaving for most part differently as compared to this particular group. And in here, there is a conformational change that happens that we have talked about. So this is a nice signal. For most part, as you can see, you can't really figure out, but if you do really fine experiment, there is a small shift in fluorescence. For example, antithrombin is exactly here, and this is blue shifted by approximately 2 to 3 nanometers. It's a small shift, not very useful, but sometimes you actually have 5 to 10 nanometer shift either on the right or on the left. And even that can be a signal. That can be a change that we can use in order to measure interactions. So the experiment is very simple, and this is what you would be doing. All the trainees would be doing, hopefully in the next camp. So the experiment is very simple. What do? You figure out, you measure the change in fluorescence as a function of concentration of the ligand under whatever conditions that you are using. And what do we find? In this particular case, where there is a fluorescence increase, as you increase the concentration of the heparin pentasaccharide under certain conditions, this is a low ionic strength condition, then you see that there is a linear increase followed by a saturation. So this can be fitted to an equation that you can easily derive, which is called the quadratic equation. You can easily derive based on simple stoichiometric binding. Protein, which is P, in this case antithrombin, binds to ligand, in this case heparin pentasaccharide, and you set up the equation, resolve it for the concentration of the protein ligand complex, And the protein ligand complex is directly, the concentration of the protein ligand complex is directly proportional to the change in fluorescence. Greater the protein ligand complex, more the change. So that's a direct proportionality. And this is a simple equation to solve. So you fit it to this equation. This is the fitted line. And if you look at this equation, what do you see? You see that the concentration of the protein is something that you already know. That is what you put in the system. So you have already defined it. The concentration of the ligand is something that you already know. This is what you actually put into the system. For every point you actually put into the system. So if you look at this term, these two terms are something that you already know. What is unknown is Kd, which is the affinity, is N. N is the stoichiometry of binding. So this really allows you also to get the stoichiometry of binding under appropriate conditions, not all conditions, under appropriate conditions. Under stoichiometric conditions, you can actually determine the stoichiometry of binding. For most part, for example, out here, for most part, this is N. We have to measure it under conditions where we can determine stoichiometry. But once you know what is N, you will be able to plug it in. So in this equation, Kd is the only unknown, and delta F max is the unknown quantity. So what is delta F max? Delta F max is simply the difference between the maximum fluorescence versus the starting fluorescence. So here, for example, you can see that this is about 33%. Likewise, this is about 25%, and things of that sort. So this gives you a very nice tool to quantitatively get Kd information. And in essence, for most part, depending on how stable the instrument is and depending on the signal counts per second that you would get, you can complete this experiment. Theoretically, you should complete it in less than half an hour. Also depends on the stability of the protein. Antithrombin is reasonably stable. It does tend to aggregate. But for most part, you can do an experiment such as this in approximately half an hour. So to do multiple experiments in a day, you should basically be done. Likewise, in this case, we have an increase in fluorescence, but there might be certain other molecules for which you actually see a decrease in fluorescence. But the principle is very similar, and you can actually utilize it to find Kd. Okay? The fluorescence signal doesn't have to be, in this case, the fluorescence signal arises from tryptophan of antithrombin. But it's not necessary that you have to have it. Number of times you may not have an intrinsic probe which is already present in the protein or on the glycan. It can be in either side. You can actually use something extrinsic such as TNS. So this is a molecule that binds antithrombin, and once it binds antithrombin, this molecule's fluorescence increases dramatically. So what happens is then once you start adding ACT, the fluorescence decreases because some of the tryptophan is released upon binding of ACT. So it gives you a very nice tool to be able to calculate with a probe that is already present in the protein or an external probe. Are you all with me? Yeah? Yeah, go ahead.
Is there only one tryptophan in the protein?
There are, not necessarily. You can have multiple tryptophans. Antithrombin has 4.
And that won't change your results at all, having multiple tryptophans that may be changed in different ways?
Yes, absolutely. Antithrombin, and it can.
Okay.
But for most part, if you have a generalized a signal, such as a composite signal, okay, which is sensitive to changes for most part throughout the protein, then any molecule that leads to binding would give you a signal. Okay, for example, in antithrombin, there are 2 tryptophans that are really fluorogenic, and there are 2 tryptophans that are not fluorogenic at all. So the signal arises from 2 tryptophans, and it is not the one that is closest to heparan binding. It's actually the one that's actually far away. So yeah, I mean, In fact, you can actually decipher as to where the conformational change is occurring. Is it close to the heparin binding site, or is it actually far away by appropriate mutations in the protein. Fluorescence spectroscopy is actually also very useful for doing kinetics, both slow and fast kinetics. And I'm not showing you slow kinetics simply because I think that's easy to understand. Anyway, we are doing slow kinetics in SPR. This is fast kinetics. This is actually stop flow, okay? And I completely forgot about the machine, the image of the machine, so that you know what stop flow is. So what I'm going to do is I'm going to tell you what the machine looks like, okay? Have you seen a stop flow machine? That's a good suggestion. Four syringes. Do we need 4 syringes? Rather than finding. Okay. All right. So here is a simple schematic for a stop flow. Okay. And in essence, what is a stop flow? A stop flow is, in essence, a combination of syringes. Shown here are 4, but let's not worry about 4. Okay. I will simply worry about 2. So eliminate all of this. And in essence, what do? You basically take your protein in one and your ligand in the second one. Okay? That's what you do. And then there is a pneumatic pump here, a piston that simultaneously pushes this so that the solution actually goes into the chamber directly. A cell, a quartz cell, which is a microcell. The volume could be as small as 20 microliters. And this particular cell would have light coming in or would have other detection means. For example, for fluorescence, there would be light coming in. So as soon as you push both of these, because of the pushing, there's a rapid mixing that happens, and the system homogenizes, and light is shining here, and the light is allowed to shine right at that time, and at time t is equal to 0, as soon as, and there's this very small delay, but at time t is equal to 0, you shine light and you start collecting light immediately in microseconds, and you collect it for sufficiently long time. Could be as long as 50 seconds, and you keep collecting it. So you actually are not following equilibrium. Okay? The previous experiment was equilibrium. You mix and you wait for at least 5-10 seconds. Hopefully that generates equilibrium, and you measure the fluorescence. Here, you are actually just injecting and then you're measuring right away. So it's a pre-equilibrium condition, a pre-steady state condition. And you mix and you start collecting. So you are basically following kinetics, okay? And it's fast kinetics. The reason why it is called stop flow is because you mix, and at that point it stops and the solution stays there. So there is a stopping device. Only certain volume will you be able to push. You can't just keep pushing. So it's not a continuous flow system. Yeah, you all get it? So that's basically the stop flow. And it really helps understand the fast kinetics of what is actually going on. And fluorescence is very useful. You can use other techniques also in stop flow, such as UV vis, if there is a change. Absolutely, you can definitely use. So, what typically happens is, you get a profile such as this, and once again I'm showing you some of the things that I have collected, and this is really long time ago. But it's basically for illustration of principle. So this is an example of antithrombin binding to heparin. I don't really know as to, well, this is pentasaccharide, and this is the tetrasaccharide. Okay, this is the pentasaccharide that we have seen, and this is the tetrasaccharide that we have seen. So this is the time at which the thing is mixed and the flow is stopped, and the system starts collecting light, 340 nanometers. It tries to measure how many photons are coming up per second. And what you find is if you do appropriate concentrations, which I like to use pseudo first ordered. You don't have to, but pseudo first ordered is really good. So under pseudo first ordered conditions, you typically get an exponential increase. In this case, antithrombin binding to heparin leads to an increase in fluorescence, right? So therefore, you would get an increase in fluorescence, and that is an exponential increase in fluorescence. So this is the real data, and this is data from one run. Normally we don't do one run. We do 10 such runs on average, so that we get a nice, much nicer profile. How do I know whether this is exponential? I know this is exponential because if I fit it to an exponential equation, the fit is reasonably good across the board. So you have very, very little residual, small residuals. So in essence, from this, the exponential increase, you can find the rate constant, the rate at which pentasaccharide, or for that matter ligand, binds to protein, the rate, the first ordered rate. Why is it? Because it's a pseudo first ordered process. And if we measure these rates at several different concentrations, then what do we get? We get something like this. So initially it is linear. So across these lower concentrations of the ligand, it is linear. But then as the concentration starts increasing, you actually start to get more and more plateauing. It tries to reach some kind of a saturation. So if the process is one-step process, you would basically get a linear profile across the board. And then this is a characteristic feature of a two-step process, that there is a complex that is formed in between, such as that. And because this complex, after a while, because this complex tends to accumulate, and it doesn't convert this into the product fast enough, that you start to see saturation. So it's a two-step process. This is step number 1, and this is step number 2. For this particular pentasaccharide, there is an increase in the rate of reaction as the concentration increases, and that subscribes to this sort of a profile. This is called induced conformational activation. It is actually induced. The conformational change is actually induced. But when you eliminate D from the pentasaccharide, when you work with this tetrasaccharide, the profile is completely different. And that profile tells us that the mechanism is not this, but the mechanism is a pre-equilibrium mechanism. So in real life, and this is the reason why antithrombin by itself is active. There's a basal activation that we have actually talked about earlier. The basal activation arises because antithrombin actually exists in the native form as well as in the activated form, and it keeps shuttling back and forth. It's just that this equilibrium lies so much to the native form that there's a very small proportion of the activated form. But by eliminating D, you have now converted into a molecule that does not recognize native antithrombin, but recognizes the activated antithrombin. So there is a complete change in the activation mechanism. And that tells you a lot about what elements are recognition features, what elements are simply affinity features, why specificity is engineered, things of that sort. Okay? Was this too complicated? Yeah.
Yeah, so when we look at this fluorescence, we're basically looking at the tyrosine part.
Tryptophan,
sorry,
yeah.
Tryptophan, right? So just by looking at tryptophan, how can you deduce the global interaction and I mean, how can you derive a mechanism just by looking at the tryptophan part of the whole antithrombin protein?
Very good question. Excellent question. So I think that the doubt in your mind is that if you have a protein such as that, and you don't know where the tryptophan is, and your ligand binds here, how can you actually know that things are happening across the entire protein? Yeah, you can't really know. Absolutely. You can't say by this one evidence that, oh, the change is global or local. It could be an absolutely local change, and we are actually following local changes. But then in combination with structures that have been made, the location of tryptophans, which tryptophan is fluorescent, which tryptophan actually gives two. So one of them gives 15% and the other one gives 18% or something like that, increase in fluorescence. And they are not really where antithrombin is. I just made that statement. They're actually far away. And therefore, those tryptophans are actually sensing something. So we know, and in combination with the enhancement in the rate and the structures, we know that this thing is actually inducing, the first change is actually happening somewhat here, followed by another change that is actually happening over the entire protein body. So we had to put things together to be able to do it. And that, I'm giving you a very simplified picture, but there have been, I would say not 1,000, but at least 100 papers published on this as to what is actually happening during the process of activation. Good question. Yeah.
Those tryptophans are urine or surface exposed, solvent exposed?
Tryptophan 49 is actually exposed. It is really involved in heparin binding, but it's not the tryptophan that is contributing to a significant extent to fluorescence. 227, those are the ones, and those are fairly inside. At least the aromatic part is fairly inside. Yeah. Okay.
Can you please educate me on this? What does this other C curve mean? The curves.
The C curve simply means that the rate of antithrombin binding to this tetrasaccharide is really high under low concentration conditions. It's actually highest when this is not there. The rate of activation is highest when this is not there. And this actually leads to a decrease and followed by a saturation. So it is a two-step process, but it actually leads to a decrease. So there might be some sequences that actually lead to a decrease. Not necessarily all sequences would lead to an increase. And it's a defect of recognition. This is a defect of recognition. This molecule is not able to recognize antithrombin. Certain GAGs may not be able to recognize native species. Yeah? Okay. Surface plasmon resonance. We have, 10 minutes. So, surface plasmon resonance is also a very well utilized technique. It does have some limitations, but obviously all techniques have limitations. But you have to be careful. If applied in the right manner, it can actually give you very good information. In addition, this is a technique that not only gives you affinity information, but it also gives you slow kinetics information, the on and the off rates. So, the principle is fairly complicated. On the application side, it's very easy to use, but understanding the principle is really a very complicated thing. For most part, what typically happens with certain surfaces of gold and silver, especially that are really, really fine surfaces, that when these surfaces are exposed to light, they typically lead to generation of waves that travel along the surface. It's really short waves. And the oscillation of these waves generates a phenomenon called plasmons. And this is altered by the change in the media that is in contact with these surfaces. So if you change the medium, simply the medium, then the angle at which this light is reflected is going to be significantly different. So a simple phenomena would be that if you load gold or silver surface, primarily gold surface, with proteins, then they would have one particular angle of reflection. But if you have protein complex to a ligand, that is a change in medium, and that could be sufficient for engineering a change in the angle of reflection. And that gives you a signal. That gives you a signal for binding. So for most part, if, for example, you start off with some value, any reference point, and then you add the ligand, then what typically happens is the binding can be followed under kinetic terms, and you see that as the molecules start coming in, more and more protein sites are occupied by the ligand. That leads to a change in the signal. RU stands for the response unit. And eventually it saturates. For most part, majority of protein binding sites are saturated. And that gives you the K-on, the on rate of interaction. Then followed by this, what is typically done is that you wash off without, let a buffer, put in a buffer without the ligand, and therefore these, whatever sites have been occupied, they would start getting dissociated, and you measure the change in refractive index once again as a function of time. And that gives you the off rate. The ratio of the two is actually the affinity. So this is a good technique for doing kinetics as well as thermodynamics at the same time. And it's very useful primarily for molecules that are able to exhibit this small change. So this is a highly specialized technique where even small change can be detected. For most part, this technique is useful for proteins, especially if they undergo some changes. And for most part, the advantage of this technique is that it is useful for a fairly large affinity range. Fluorescence has a limitation. Under certain, if the affinity is too poor, obviously you'll have a very weak signal. Or fluorescence also has a problem of photo bleaching that I didn't really talk about, or inner filter effect and things of that sort, where if it is too much, then that creates a problem. Here, this is a fairly wide range, and therefore it's very well utilized. Kinetics can be measured. For most part, we can use buffers that are physiological and therefore pretty good. And what typically happens is that the cell is very small, and therefore you can get away with microgram quantities of your biomaterials. Limitations are that you can typically use ligands that are sufficiently large. So if you really have a very small molecule, molecular weight 300, for most part you don't get a signal. Larger molecules, heparin for example, gives you GAGs, gives you fantastic signals because typically they are very long. Pentasaccharide has been done, but the effect is not as great. The idea has been that you can couple the glycan to the gold surface rather than coupling the protein to the gold surface. So then you would have protein, which is a much bigger molecule, coming into the ligand and therefore giving you a bigger surface. There are pros and cons to both of the ideas. Okay. You have to be careful that when you couple onto the gold chip, you leave the site of interest, your binding site, exposed. So normally coupling is typically run in the presence of the ligand, so that you don't have that. And many times, because that we all want to do things really fast and be done with it, we are not particular about how we, what sort of coupling do we get. There can be numerous inhomogeneities of coupling, and therefore you might actually get KD. The other challenge is that a number of times you might have mass transfer effects limiting the role. And in terms of GAG, binding to proteins, it has been observed that the surface plasmon resonance typically gives much higher affinity than what is measured in solution. So those are issues. Those are issues that I think I'm not going to talk about ITC at all. We'll simply end at surface plasmon resonance. and hopefully when you all come next time we'll do ITC and things of that sort, and he might have time. Very good. So okay, so lower loading densities could also be a problem. All right, so this is where I would probably end. So this is affinity versus avidity, and this is primarily in relationship to glycans, where clustering is a major effect. So affinity is one molecule binding to one molecule, and that is intrinsic affinity. Okay, avidity is what glycan people tend to use, that because there is clustering, whatever affinity that they actually measure, they call it avidity, because it's not really true affinity. So it's measured affinity, but it is, the term that is used is avidity because there is clustering. So it actually refers to multi-site interaction, and it's primarily for lectins, because that's not really the case. In fact, as we have seen, it's pretty dramatic, and what typically happens is that, and this is deducible primarily from the SPR signals. So here is a monomer. M stands for monomer. This is a dimer, and this is one of the lectins. SSA is one of the lectins, and it recognizes 6' lactose and lacnac. So you can see that the monomer gives a very poor response in terms of SPR, the on rate, as well as a fairly fast dissociation. It comes off fairly fast. The dimer actually gives you a much higher response, and the dissociation also is then reduced. Likewise, under several different conditions, under competitive effects, you do the same thing. So affinity and avidity are two different things, and avidity primarily refers to the clustering effect, and therefore for most part people do not measure affinity per se because of this oligomerization. Okay, and avidity is what you would see in the literature. Okay, let's stop here because we can keep going on and on. Questions? No questions? All right, very good.